Good evening, everyone. So today I learned about the osmotic pressure. This channel is basically of I have learned something, so I want people to listen what I learned, or if you want to understand, you can understand what I learned. So this is osmotic pressure, and osmotic pressure only depends upon the cell wall of the cell, mainly the prokaryotic cell, which has a single celled morphology is maintained by the cell wall, because the prokaryotic cell have the cell wall and some of the eukaryotic cells have the cell wall, and in cell wall protects the cell from changes in osmotic pressure. Osmotic pressure occurs due to the difference in the concentration of solute on opposite side of the semipermeable membrane. So the semipermeable membrane is nothing but the cell wall. The solute is the one which is of larger quantity, like, and the osmotic pressure occurs due to a difference in the concentration of solute, which we will further learn in the next discussion. What does the osmotic pressure depends upon? The external environment, and there are three types: isotonic, hypertonic, hypotonic. Isotonic means, as you know, in the word iso it means equal. The concentration is same both on the inside as well as the outside of the cell. And in the hypertonic, the concentration of solute is more than the inside the cell. Therefore the water diffuses out of the cell and into the external medium. If I want to make it easy to understand, then let's take a let's think that the cell is present, like the potato, right? We have learned in so many of our 9th and 10th class the potato experiment, that the potato is kept in the water, so the concentration of water is more than of the inside of the cell. Therefore what happens, the outside membrane comes inside of the cell, which makes the water to come out of the cell. So in this, the word diffuse is used because we have to explain it in the manner of the microbiology, right? I mean, how I'm explaining, we can't write in the paper. Actually we can write, but it does not give us the same amount of marks which we are expecting. So what is hypertonic medium? It's the opposite. Hyper means, you can see that hyper means what? It's generally, it means higher. Okay, so already, as I told, it is all is depend upon the cell wall, and the osmotic pressure occurs quite the outside of the semipermeable membrane. So it everything depends upon the outside concentration. In the isotonic, the outside concentration is same, and in hypertonic, the outside concentration is more. Okay, I'm sorry for the disturbance of the outside sound, because there are some construction going near our home. Now, hypertonic medium. Hypertonic means, as you know, hyper means higher, hypo means less. The concentration of cell is more than the solute. Therefore, the water will move by osmosis into the cell. This causes the swell and potentially lies and burst. Okay, so as you know, this sentence is really very confusing. It was also confusing to me at the beginning. We can also write that the concentration of the solute is less than the concentration of the cell, but here they have opposite, right? You can write it, no problem, because it is the same. Therefore the water will move by osmosis. Here you can means write, note down that the word osmosis is very important in the hypotonic, because we are learning about the osmotic pressure. So there are certain words which are very important to be noted in the sentence, like the diffuse and the osmosis in the cell. This cause the sweaty potentially burst. Now, why these two words are important? Because the examiner, while they are correcting, they will not read the full sentence. They will search for certain words which are used, which are very necessary for them. If these are not there, but your sentence is correct according to the definition, then they'll give you half mark, because but you have not come to their expectation, then they can cut your marks. That's why it's so necessary for you all that you have to do like get the same. You can use the word. Bye-bye. Sorry, because I'm not habituated of teaching people. Okay, then the degree to which a particular cell is able to withstand changes in osmotic pressure is called tonicity. Now, let me just tell you. In hypotonic, as we know, by the osmosis, the water will go inside. Okay, so what happens if let us take the example of a fruit, that's tomato. We know that if it is kept in the fridge for longer time, it slowly starts rotting. Why? Because the water contained inside the tomato increases. Okay, so for certain, there is certain period, right, to control the anger or to control the pressure. Therefore, to control the degree to which the particular cell withstand changes in osmotic pressure is called as tonicity. It is this tonicity means the what, as you know, the boiling point is 100 degrees Celsius, right? That's why it is known as the boiling point. This is the same. It is the time when the cells can withstand the osmotic pressure. Now, cell wall can withstand the subtle changes in osmotic pressure and maintain their shape. The cell wall is the major role. They have the major role in the osmotic pressure, because they use in osmotic pressure as well as they are used for the shape of the cell. The primary function of cell wall is to protect cell from harsh condition. Obviously, because cell wall is a layer of protection. Like the egg cell, like the egg has its own shell, like the cell also have its own cell wall to protect it from the harsh condition. Next, the major component of bacterial cell wall is called what? Peptidoglycan. This is very important to know, because in some of the viva question also they ask what is the major component of the cell wall. They will say peptidoglycan. Here murin is used because the peptidoglycan contains the murin acid. Okay. The major component of bacterial cell wall is peptidoglycan. The major component of bacterial cell wall is peptidoglycan. I'm so sorry because I have practiced it. Then each layer is composed for N-acetoglucosamine and N-acetylmuramic acid, which is known as NAM and NAG. We have to remember this, obviously. The NAG layer and the NAM layer, which see how these are made. The peptidoglycan layer, how it is made, is the NAG N-acetylglucosamine and the NAM N-acetylmuramic. In between them, the peptide bridge is they both are attached with the peptide bridge actually. Therefore, the peptidoglycan forms. The peptidoglycan subunits are made inside the bacterial cells and they export and assembled in layers, giving the shell its shape. This means that the peptidoglycan generally forms inside the cell and then after they are exported outside the cell to form its shape. What we know about peptidoglycan? Okay. First, it is a cell wall. It is means where the cell wall is made. Then it's made inside and then exported outside the layer to give the cell a shape. And peptidoglycan is unique in bacterial cells, and its antibiotic drugs are designed to weaken the cell wall and make bacterial cell more susceptible to the effect of osmotic pressure. We all know that in gram staining also there are some components which are used to break the cell wall, not to break the cell wall, to give its color. And also there are some bacteria, some drugs are used to break the cell wall or to weaken the cell wall. In human immune system recognized bacterial pathogens by detecting peptidoglycan on the cell wall surface of the bacterial cell. These cells engulf and destroy the bacterial cell using enzyme called lysozyme. This means what? The human immune system. it has the power to recognize the pathogens, so they recognize the pathogens by detecting the peptidoglycan outside the bacterial cell, and these engulf and destroy the bacterial cell by using the enzyme which is known as the lysozyme, which breaks down and digests the peptidoglycan. Now in gram staining also, different two types of cells. Why the gram staining is used? Because they use for the two common types of cells. Peptidoglycan layer are commonly embedded with TAS, teichoic acid carbohydrate chain. TA is thought to stabilize peptidoglycan by increasing its rigidity. Teichoic acid, you can remember, and in the next topic we'll learn about the gram staining. And I hope that I have made you understand about the osmotic pressure. If any doubt is there, you can ask. Basically this video I'm making to, like, when I'm in my exam hall, I listen to my friends advice, then listening to the teachers or reading the notes. So I try my best to make you understand why what I understood by this topic. Thank you.